Hello everyone, I'm Jing Han and I'm a research fellow from Duke NUS Medical School, Singapore. So I got my PhD from the National University of Singapore. Today, I would like to discuss the genomic and epigenomic landscape of cholangiocarcinoma, and this is on behalf of my supervisor, Prof. Tay Bin Tin, who is unable to make it down for the presentation today. Cholangiocarcinoma is a malignant tumor that arises from the epithelial cells lining the bile duct. There are two major classifications of cholangiocarcinoma: the intrahepatic carcinoma and the extrahepatic carcinoma, depending on the anatomical position on where the cholangiocarcinoma arises. Intrahepatic carcinoma arises from the bile duct within the liver, whereas extrahepatic cholangiocarcinoma arises from the distal or the ileum region. The cholangiocarcinoma is the second most common form of liver cancer, and it accounts for about 10 to 25% of all liver cancer cases. The risk factor for cholangiocarcinoma differs by geography. So for the Western places, the major risk factor are cholangiotitis, hepatitis, whereas for the Asian regions, in particular Southeast Asian, infection by liver fluke is the major driving force for cholangiocarcinoma. Cholangiocarcinoma has been reported in many different countries worldwide, but it is particularly prevalent in the Asian region, including China and South Korea. So the prevalence of cholangiocarcinoma is very high in the northeast region of Thailand, where an estimated 20,000 cases of fluke-positive cholangiocarcinoma are reported each year. Infection by liver fluke of this species, which is called OV in short, is a very major public health issue in East Asia, especially so in Southeast Asia, because OV fluke is endemic in these regions, which includes Vietnam, Cambodia, Laos, and Thailand. One of the hosts for OV fluke is freshwater fish, and these fish are caught and consumed by the local people. The traditional way where these local people consume the fish is raw or undercooked. And what happens is that once the fish is consumed, the mature larvae will travel to the gastrointestinal system, exist in the duodenum, and from there travel to the biliary duct. In the biliary duct, the mature larvae will then develop into the adult form of fluke in about 1 month's time. The adult fluke will then live in the biliary system, where it causes chronic inflammation. It disrupts the metabolic environment of the host bile duct. It secretes the fluke-associated growth factor, and all these factors together are thought to drive fluke-associated cholangiocarcinoma. In order to understand how OV fluke is able to survive so well in a harsh environment such as the bile duct, we have sequenced the genome of the OV fluke using the multiple displacement PCR strategy. When we analyzed the genes in the genome, we surprisingly found that many of the genes are involved in nutrient metabolism. So this suggests that OV fluke is able to assimilate the nutrients from the bile duct and then utilize it for its own energy needs. What's more interesting is this cholesterol metabolism pathway. We found that for the cholesterol transporters, OV fluke contains of many copies of these transporters. An example, MPC2, OV fluke alone have 52 copies of this gene. So these transporters transport the LDL form of cholesterol from the cells to the endodermal system so that it can be hydrolyzed to the free form of cholesterol. The free form of cholesterol is the cholesterol that is easily used by OV fluke for its own energy needs. Thus, so far, we can see that many differences affect cancer initiation and development. So this includes geography, diet and culture of people, exposure to carcinogens and pathogens. In order to understand how these factors separately and together regulate tumorigenesis epigenetically and genetically, our group has collected close to 500 cholangiocarcinoma tissues from different geographical regions and of different etiologies. We have performed whole genome sequencing, targeted sequencing, exome sequencing, and then DNA methylation array, as well as look at gene expression using microarray or RNA-seq. We analyze these data sets separately and also integrate them together to analyze so that we can more precisely subtype the different cholangiocarcinoma cases of diverse population molecularly. From our genomic studies, we have found many cholangiocarcinoma driver genes based on the prevalence of the mutation in our cholangiocarcinoma cohort. What is most striking to us is the differences between the driver genes that drive Fluke positive and Fluke negative cholangiocarcinoma. From this chart over here, we can see that SMAD4, RE1A, P53 are almost equally associated with Fluke negative and Fluke positive cholangiocarcinoma, whereas genes such as IDH1 and PBRM1 are more associated with Fluke negative cholangiocarcinoma, while KRAS mutations and PTEN mutations are more associated with Fluke positive cholangiocarcinoma. In addition, from our genomic profiling, we are able to identify new potential tumor suppressor genes. So they are MET2K4, STK11, and RASA1. From these schematic cartoons over here, we found that most of the mutations of these three genes are of the truncating inactivating mutation type. And most of the mutations are also localized to the important domains. So significantly, STK11, they are located in the kinase domain, which is important for the function of STK11. We validated the tumor suppressor potential of one of the genes, RASA1, in vitro in cholangiocarcinoma cell lines. As can be seen here, M213 is a Fluke positive cell line, whereas HUCCT1 is a Fluke negative cell line. We knock down RASA1 using shRNA, and once the expression of RASA1 is knocked down, we find that the migration and invasion capability of Fluke positive and Fluke negative cell line increases, suggesting that indeed RASA1 is a strong tumor suppressor consistent with its purported role. One of the main aims of our study is to identify new potential therapeutic targets for cholangiocarcinoma patients. And therefore, we are interested in examining clinically relevant pathways. One such of the pathway we found is driven by the FGFR fusion proteins. From our cholangiocarcinoma cohort, we found many different forms of FGFR fusion proteins, and the fusion partners are from different chromosomes. Together with other groups, we have confirmed that FGFR fusion is a major driver of cholangiocarcinoma tumorigenesis. When we examined further, we found that, interestingly, the FGFR fusion appears to be more commonly found in non-fluke associated cholangiocarcinoma compared to Fluke positive cholangiocarcinoma. In contrast, mutations in the kinase pathway, they are more associated with Fluke positive cholangiocarcinoma compared to Fluke negative cholangiocarcinoma. This is significant because there have already been 2 FDA-approved drugs for the FGFR2 fusion protein. So the first drug here has been approved last year. The second drug in the table just approved several months ago. And the third drug is still an investigational drug, but has already been awarded breakthrough therapy designation by FDA. So our study shows that these drugs are not effective for the Fluke positive cholangiocarcinoma cases, but more effective for the Fluke negative cases with the FGFR2 fusions. Other interesting targetable pathways that we have found are the ERBB RAS pathways. So the mutations in genes of this pathway make up close to 17% of all cholangiocarcinoma cases in our cohort. When we perform a literature search for drugs that target this pathway, we found that indeed there are many drugs that target the members of this pathway, and many of them, for example, those in orange, they are FDA approved, but they are FDA approved for other cancers, not cholangiocarcinoma. Our study shows the potential of repurposing this drug for cholangiocarcinoma patients with mutations in this pathway. The next pathway, which we found interesting, is the DNA repair pathway. In fact, there are many DNA repair genes that are mutated in cholangiocarcinoma, suggesting that there's genomic instability, and therefore drugs such as the PARP inhibitors, which is a DNA repair protein, might be useful for cholangiocarcinoma. In fact, there have already been several clinical trials, basket clinical trials, to test the efficacy of these drugs for cholangiocarcinoma cases. We also found mutations in the Wnt signaling pathway, so they add up to about 20% of the cholangiocarcinoma cases in our cohort. Again, from literature search, we found many drugs that target members of this large Wnt signaling pathway. And these drugs are either in preclinical or clinical development. But again, our study suggests the effectiveness of using these drugs for the cholangiocarcinoma cases with mutations in this pathway. Very significantly, we found many epigenetic modifiers mutated in cholangiocarcinoma. So this makes up close to 50% of our cholangiocarcinoma cohort. Of all these epigenetic modifiers, we are most interested in BEP1, in which we performed an extensive follow-up study on. So in addition to its role as an epigenetic modifier and DNA repair, BEP1 plays roles in many cellular processes in the cell, as revealed by this cancer discovery paper last year. So this includes role in metabolism through oxidative phosphorylation, role in apoptosis, as well as ferroptosis. We found BEP1 frequently mutated in cholangiocarcinoma. That is about 8 to 10% of the cholangiocarcinoma cases that we see. And then we knock down BEP1 using siRNA in Fluke negative cell line. So EGI1 and HUCCT1, they are Fluke negative cell line, as well as the normal cholangiocarcinoma cell line. By knocking down BEP1, we observe that the rate of proliferation and viability increases. Whereas when we overexpress BEP1, the rate of proliferation and viability decreases. So this shows that BEP1 is a strong tumor suppressor in cholangiocarcinoma. Through our genomic study, we are also able to identify a potential new risk factor for cholangiocarcinoma, that is exposure to aristologic acid, or AA we call in short. AA is predominantly found in the Aristolochia species of herbal plants. So there are many different forms of AA. So far, only AA1 and AA2 have been shown to be strong mutagens. So all these herbs contain AA, and these herbs are commonly consumed as herbal remedies, especially in the East Asian countries. AA has been classified as a potent group 1 carcinogen, and together with other groups, we have found that exposure to AA increases the risk of nephrotoxicity as well as upper urinary tract urothelial carcinoma. As compared to other mutagens that induces mutation in DNA, AA is able to induce the most number of mutations even as compared to exposure to UV light and as well as exposure to cigarette smoke. We found that AA causes a predominance of A to T, T to A transversions, as can be shown here, on the non-transcribed strand of the genome, as shown in these two charts here, the S. AA causes a unique mutation signature only in the context of this unique motif. And therefore, because of the association with AA with tumorigenesis, AA signature has been assigned number 22 mutation signature in the Cosmic Mutation Signature database. Interestingly, we found that by treating cells with the purified form of AA, we are also able to induce AA mutation signature in vitro. We found a number of cholangiocarcinoma tumors with the AA signature in our cohort. And interestingly, these tumors are predominantly from Taiwan. So this shows the contribution of geography towards driving distinct molecular subtypes of cancer. Finally, we integrated our genomic study with genome-wide DNA methylation data. We were quite surprised to find two major DNA methylation subtypes in cholangiocarcinoma, cluster 1 and cluster 4. Cluster 1 is made up predominantly of Fluke positive cholangiocarcinoma cases, and we find that there's increased island hypermethylation as compared to shore hypermethylation. Whereas cluster 4, which is made up of predominantly Fluke negative cholangiocarcinoma, has increased shore methylation compared to island hypermethylation. We found that the hypermethylation pattern could be driven by distinct methylation mechanisms. So for cluster 1, there's reduced TET1 expression and increased EZH2 expression, which are known to be associated with DNA hypermethylation. Whereas in cluster 4, because they are all non-fluke associated cholangiocarcinoma with IDH and BAP1 alterations for majority of the tumors in these clusters, and activating mutations in IDH1 have already been shown to cause hypermethylation in many different cancers. And BAP1 mutations have been associated with hypermethylation in RCC, and now we show that it is also associated with hypermethylation in CCA. We went on further to analyze the mutational signatures between cluster 1 and cluster 4 of the tumors, and we found that signature 1 is highly enriched in cluster 1. So signature 1 is represented by the spontaneous deamination of hypermethylated CpG to TpG. So the question that we have is whether the hypermethylated cytosines in the island of cluster 1 substrates for signature 1 mutations. So we integrated where the DNA is mutated with the methylation status, and indeed we found that the cytosine in these TpG islands are spontaneously deaminated for cluster 1 tumors. So for cluster 4 tumors, there are also many different, there are also a high number of hypermethylated cytosine, but these cytosines are not spontaneously deaminated into TpGs. Based on all our results so far, we propose that these differences are consistent with a model where early in tumorigenesis, cluster 1 cholangiocarcinoma are likely to be driven by external carcinogenic agent with early epigenetic deregulation, while cluster 4 cholangiocarcinoma are driven by pioneer genetic events such as mutations in IDH1 or BAP1, and the epigenetic aberration arises as a downstream consequence. In our model, Fluke infection induces chronic inflammation, disrupts the metabolic environment of the host bile duct, secretes Fluke-associated growth factors, and induces genome-wide epigenetic deregulation. The cytosine residues then experience aberrant methylation and are at a higher risk of spontaneous deamination and mutation, which explains the enrichment of cluster 1 mutations in this subtype. And because the process of hypermethylation, deamination, and mutation are inherently stochastic cell to cell, such events will lead to increased levels of intratumor heterogeneity. Whereas for cluster 4 cholangiocarcinoma, which are Fluke negative, somatic mutations in chromatin modifier genes such as IDH1 and BET1 occur as the initial primary event that precedes epigenetic deregulation, and it drives rapid clonal outgrowth, directly inducing DNA hypermethylation. So far from our integrated analysis, we are then able to identify 4 cholangiocarcinoma subgroups with distinct therapeutic relevance. Of these 4 different subgroups, we are most interested in cluster 3, which have the highest copy number alterations and increased expression of immune-related genes. We analyzed the enhancer activity of the cluster 3 and found that for the enhancers that regulate the expression of immune genes, they are also highly active. In order to study immuno-oncology in cholangiocarcinoma, we performed nanostring for our cholangiocarcinoma tumors, so as to better look at immune activity and to quantitate immune cell subsets. Interestingly, we found that AA-associated cholangiocarcinoma has increased expression of immune genes and enhanced immune pathways with increased number of immune cells as well as immune cell populations, suggesting that the AA-associated cholangiocarcinoma tumors are immune hot. This is consistent with recent publications on liver cancer, where AA mutations are thought to increase the immunogenicity of the tumors as well. In terms of tumor inflammation score, consistent with the previous slide, AA-associated tumors tend to have higher inflammation score as compared to non-AA-associated cholangiocarcinoma. So in conclusion, our study shows that many different factors, including environment and geography, can together generate distinct molecular subtypes of cancer. This distinct molecular subtype of cancer will require different tailored therapies, and studies that integrate omics dataset, multi-omics dataset, so as to analyze whole genome and epigenomics of cancer from diverse population can help to identify new driver genes, non-coding promoter mutations, and structural variants, which are very important for drug discovery. So thank you, everyone. We would like to acknowledge our colleagues as our collaborators and our funding agency, because without them, this study will not be possible. Thank you. So any questions?
So thank you so much, Dr. Han, for joining us today. And please, if you have any question, feel free to submit it on the chat. Okay, so thank you so much, and hope to see you soon in next month for a new seminar. Thanks, Dr. Han.
Thank you.
Bye-bye.
Bye-bye.